Everyone, today we are going to discuss about the topic that is control of microorganisms, that is physical and chemical methods, and also about the staining of microbial cells. Let's move on to the topic. We know that many organisms are beneficial and necessary for human well-being, but the control of microorganisms is essential in order to prevent the transmission of diseases and their infection, stop decomposition and spoilage, and prevent all unwanted microbial contamination. The main purpose of this control of microorganisms is to destroy the pathogens and prevent their transmission, and also to eliminate these microorganisms that is responsible for the contamination of water, food and other substances. So these are the two main purposes for controlling the microorganisms. So microorganisms are controlled by several means of physical and chemical methods. Physical methods include such as high or low temperature methods, desiccation, radiation, filtration, like that. So in this in this video we will concentrate more about the physical and chemical methods to control this microbial growth and also about some staining techniques. Let's see some main methods to control of microbial growth. The first method is sterilization. Sterilization is the process of removing or destroying all forms of microorganisms from a product using a suitable agent. So, a sterile object is free from all sorts of microbial life forms including endosperm, the topic that is control of microorganisms, that is physical and chemical methods, and also about the staining of microbial cells. Let's move on to the topic. We know that many organisms are beneficial and necessary for human well-being, but the control of microorganisms is essential in order to prevent the transmission of diseases and their infection, stop decomposition and spoilage, and prevent all unwanted microbial contamination. The main purpose of this control of microorganisms is to destroy the pathogens and prevent their transmission, and also to eliminate these microorganisms that is responsible for the contamination of water, food and other substances. So these are the two main purposes for controlling the microorganisms. So microorganisms are controlled by several means of physical and chemical methods. Physical methods include such as high or low temperature methods, desiccation, radiation, filtration, like that. So in this video we will concentrate more about the physical and chemical methods to control this microbial growth and also about some staining techniques. Let's see some main methods to control of microbial growth. The first method is sterilization. Sterilization is the process of removing or destroying all forms of microorganisms from a product using a suitable agent. So, a sterile object is free from all sorts of microbial life forms including the endospores and viruses. It is mainly adopted for the decontamination of instruments, surfaces that comes in contact with the microbes and thereby helps in preventing their spread. So this is about the sterilization. Next, about the disinfection. So it is the process of elimination of all or most of the disease causing microorganism from an object. So an agent is used for this purpose. It is called disinfectant. When disinfectants are formulated for the use on living cells like skin surfaces, then they are called antiseptics. So antiseptics are commonly used to decrease the microbial populations on our skin surfaces like that. So these are some main methods to control of microbial growth. Next, about the physical methods for the control of microbes. So some common physical methods used for controlling microbial growth include the application of high temperature, that is heat, desiccation, filtration, radiation and ultrasonization, etc. First one, heat sterilization. It is the most widely used and reliable method of sterilization. It is a temperature-dependent sterilization techniques where exposure of cells to high temperature leads to the destruction of cellular enzymes and proteins, thereby leading to the cell death. So here the heat kills the microbes by denaturing their enzymes like that. The efficiency with which heat can inactivate the microorganism is depend upon the degree of heat, the exposure time and the presence of water. For that, some parameters are used. They are thermal death point, thermal death time. The thermal death point of a microorganism is the lowest temperature at which all the microorganisms are killed within a 10-minute exposure time. Whereas the thermal death time is the length of the time needed to kill all the microorganisms in a sample at a given temperature. So these are the parameters used in the heat sterilization, that is thermal death point and thermal death time. And this heat sterilization can be done using two methods. They are dry heat method and moist heat method. Dry heat methods are of three types. They are flaming, incineration, hot air oven. And moist heat method includes pasteurization, boiling, steaming, autoclave like that. Autoclaving. It is a sterilization method utilizing pressurized steam which effectively removes all the microbes, even the spores. So this autoclaving is carried out by an instrument called autoclave. So it is an instrument that maintains a high temperature of 121 degrees Celsius at 15 lbs per square inch pressure. That is for holding time for minimum 15 minutes. So all the cells and endospores will be killed within this time. This is about autoclaving. Next is about pasteurization. Pasteurization is a form of microbial control for food products and that are easily affected by higher temperature. It was developed by Louis Pasteur in the 1860s. And it is the method for preventing the spoilage of wine and beer. But nowadays it is commonly used to kill the heat sensitive pathogens in milk and other food products. Pasteurization are of mainly three types. They are low temperature hold method, high temperature short method and ultra high temperature method. First one, high temperature short time pasteurization. In this method, it exposes the milk to a temperature of 72 degrees Celsius for 15 seconds, which lowers the bacterial numbers while preserving the quality of milk. The other method is ultra high temperature pasteurization, where the milk is exposed to the temperature of 138 degrees Celsius for 2 or more seconds, which preserves the milk for longer shelf life even without the refrigeration. But in the lower temperature method, the milk is exposed to the temperature of 63 degrees Celsius for 13 minutes before it cooled to 7 degrees Celsius. So these are the three different methods of pasteurization. So these are the some methods comes under the moist heat sterilization techniques. So the next physical method used for controlling the microorganism is filtration. Filtration is a method providing physical removal or exclusion of all the cells in a liquid or gaseous medium. So in this picture you can see the filtration process. For the filtration we use a membrane filter. You can see a membrane filter in this picture. So this membrane filter provides a useful way of sterilizing materials such as vaccines, antibiotic solutions, animal sera, enzyme solutions, vitamin solutions and other solutions which may be damaged or denatured by high temperatures or chemical agents. So this is about the filtration. The next method is radiation. There are various forms of radiation that can be used to kill the microbes or inhibit their growth. Its efficiencies depend on the wavelength, intensity, duration, like that. Radiations can be two methods. They are ionizing and non-ionizing. Ionizing radiations are highly penetrative and that can cause direct damages to the DNA and that leads to the cell death. Because of their high penetrative power, ionizing radiation is used to sterilize packaged materials, non-autoclavable materials such as petri dishes, disposable plastic inoculating loops, gloves, and other heat-sensitive materials and some medical equipments like that. Non-ionizing radiations, which is less penetrative, and used to control microbial growth as they induce indirect damages to the DNA through the formation of reactive groups within the cell. So they are less penetrative and use less energy than ionizing radiation. So ionizing radiations are highly penetrative and it causes direct damages to the DNA. But non-ionizing radiation, they are less penetrative comparing to the ionizing radiation, and also it occur indirect damages to the DNA. Next is about desiccation. It is the method of drying or decreasing the water content of products so that the microbial growth is inhibited. And this method is largely adopted in food industry as it keeps a check on microbial multiplication and prevent the food spoilage. The last one is ultrasonization. This is a method that utilizes sound waves of ultrasonic frequencies that is greater than 20 kHz for controlling the growth of microorganisms. It generates the alternating low pressure and high pressure waves in liquids and leading to the formation and violent collapse of small vacuum bubbles. So this is about the ultrasonization. So in physical methods, we discussed about the heat sterilization. They are of two types, dry heat method and moist heat method. And most heat methods includes autoclaving, pasteurization, steaming, boiling, like that. And some of other physical methods are desiccation, radiation, filtration, ultrasonization. Next is about the chemical control of microbes. A wide varieties of chemicals are employed in the control of microorganisms. So some of the chemical agents commonly used for the control of microorganisms are alcohols, aldehydes, heavy metals, stimulants, fumigations, like that. First is about alcohols. Alcoholic compounds like ethanol, isopropanol are wide and some other alcohols are widely used as disinfectants. They act by denaturing the proteins and disrupting all the cell membranes. Next is about aldehydes. They include one of the most effective disinfectants. They are antimicrobial. They inactivate proteins by forming a covalent links between the functional groups. Some of the examples are formaldehyde, glutaraldehyde, etc. Formaldehyde preserves the biological specimens, where glutaraldehyde sterilizes the hospital instruments. And they are most effective of all chemical. They are one of the most effective disinfectant. Next is about halogens. Iodine, chlorine, iodophos are some common halogens used. They can be used alone or in compounds. Sodium hypochlorite is a chlorine compound that commonly used in bleach. And iodine tincture was one of the first antiseptics to be used. Next is about heavy metals. Heavy metals like silver, mercury, copper, selenium, zinc, etc. have antimicrobial action. Even very low quantity of these elements are found to effectively control microbial growth, which is utilized in several disinfectant products. And some other chemical agents used in controlling microorganisms are sterilants, phenols, fumigations, like that. And fumigation is the process of gaseous sterilization done in an enclosed area by introducing some chemicals that will destroy the microbes in the air and surfaces like walls, floors, etc. And sterilants are the chemicals applied to inanimate the objects which kill all the microorganisms including endospores and viruses. And they can be used to treat heat sensitive critical items. Next we will discuss about the staining of microbial cells. We know that bacteria are all microscopic organisms. They are also colorless for most of the part. So in order to visualize them to study their structure, shape and other structural characteristics, it becomes necessary to make them more easily visible. So for their easy vision, the cells need to be stained. So a stain is a dye that is used to color the living or dead organelles. So mainly stainings are of two types. They are positive staining and negative staining. In positive staining methods, the actual cells are themselves colored and appear in a clear background. And they are of two types, that is simple staining and differential staining. In negative staining, the cells remain clear, that is they are uncolored, and the background is colored to create a contrast to aid in the better visualization of the image. So simple staining is the method which is using only one dye. So here a single stain is used to color the microbes. This is mainly adapted to study the morphology and spatial arrangement of the cell. Here the basic dyes are used in direct stain and acidic dye is used in the negative stain. The most commonly used stains are methylene blue, dilute carbolphetazine, safranin, crystal violet, etc. So this is about the simple staining. Next is about the differential staining. This is also comes under the positive staining method. It is a staining technique used to differentiate between microbial groups. And this method is taking the advantage of variations in chemical structures, especially in the cellular components of different bacterial groups, which led to the differential staining property. So one of the most commonly used differential staining technique is gram staining technique. Let's see what it is. Gram staining is based on the differences in the cell wall. So it is mainly based on the composition of their cell wall. Gram-positive organisms contain a cell wall that is made up of peptidoglycan, whereas gram-negative organism, their cell wall is mainly made up of lipopolysaccharide. So in gram staining, mainly two stains are used. They are primary stain, which is crystal violet in color, and secondary stain, that is safranin. So after the staining procedure, gram positive cells appear in purple color and gram negative cells in red color. Let's see how the gram staining method takes place. First, the cells are treated with a primary stain which is in violet color. They are called crystal violet. So in the first figure you can see the primary stain, that is crystal violet. So the cells are treated with this crystal violet, and this crystal violet stain all the cells within the minute, and the excess stain is then washed off. In the second step, this smear is treated with grams iodine solution which act as a mordant that increase the affinity of cellular components for the dye. So here the iodine combines with the crystal violet to form a crystal violet iodine complex which decreases the solubility of dye. So in the second figure it shows the application of the mordant, that is iodine. And the third step, you can see the smear is then treated with 95% of alcohol, that is the decolorizing agent, and immediately rinsed. And this alcohol dehydrates the gram positive cell wall and that traps the crystal violet iodine complex within. But in gram negative cells, it dissolves the lipid rich outer layer of the cell wall, thereby making it porous. Hence, the gram negative bacteria readily lose the crystal violet iodine complex, thereby decolorizing the cell. So, in the third figure, decolorization takes place. Hence, the gram negative bacteria lose the crystal violet iodine complex, and the gram positive bacteria traps the crystal violet iodine complex. And the last step is the application of safranin. So finally, the counter stain safranin is applied, which imparts a red color to the colorless gram negative bacteria. So in the last figure you can see the crystal violet iodine complex. That shows the gram positive cells, bacterial cells, and the red color cells shows the gram negative bacterial cells. So this is how the gram staining technique takes place. This is all about the staining of microbial cells. So in this overall video, we mainly discussed about the physical and chemical methods used to control the growth of microorganisms, and physical methods such as sterilization, disinfection, sanitization, radiation, ultrasonization, etc. And also the chemical methods used to control are chemical agents that are used to control are aldehydes, alcohol, phenols, like that. And also we discussed different types of staining techniques. They are simple staining, differential staining, like that. Thank you.